Today we are looking at osmosis using one type of plant tissue. We are using pieces of potato, Irish potato, for this particular experiment. Now, what we've done here, we have three aspects or three different experimental conditions set up. We are going to put three of our pieces of potato, which we have in these petri dishes. We are going to put these into a concentrated or very strong salt solution. And we are putting these into boiling tubes C1, C2, and C3. The next set, we are using a dilute solution of salt, and we are going to place our potatoes. Again, we have them all laid out here in the petri dishes. We are placing them into the same volume of solution. So again, we are using 10 mils of solution that we are going to pour into each of these boiling tubes that are labeled D1, D2, and D3. So these would be the setup for the dilute salt solution. And finally, over here, we are going to use distilled water in our final batch of potatoes and put them into, again, 1, 2, and 3 boiling tubes. So in order to make these potato strips, we have used a cork borer and obtained approximately 2 cm in length of each one of our potato strips, and then we just dried them out and kept them fairly dry by placing them on tissue paper inside of each of the petri dishes. So we're going to do this setup for the concentrated solution by pouring approximately 10 mils of this into each of our boiling tubes. Use the meniscus to ensure that we are getting the same volume into each of our tubes. Right, to avoid mixing of the solutions, I'm using 3 different measuring cylinders, one for each of the solutions that I'm using in this experiment. So I'm finished now with using the cylinder to measure the concentrated salt solution. And the next one that I'm going to measure would be the dilute solution. I'm using a different measuring cylinder for that. And the same volume that I used for doing the concentrated solution, which is the 10 mils. Right, so now I'm finished with my measuring cylinder for measuring the dilute solution. So I'm going to place that to the side just so that I don't mix up the solutions. And the final set of boiling tubes, into those I am going to also pour 10 mils of this distilled water. That aside. Next, now I have laid out all of my potato strips that are supposed to go into the different boiling tubes. So the one that I have open right now are the potato strips that are supposed to go into the tubes containing the concentrated solution. So I'm going to have to make sure that it goes down into it. Next, I'm putting in my potatoes into the dilute solution. And finally, I'm going to put these into the distilled water. Right, so now I'm going to just move these aside, and here we have our 9 boiling tubes set up, so that when we come back after approximately half an hour, we're going to leave the potato strips in here in all of these different solutions, so approximately half an hour. And when we come back, we're going to take them out, put them back in on the pieces of tissue and allow them to dry out a little bit by patting them dry, and then we're using our rulers to get measurements of the length and the diameter of the potato strips at the end of our experimental period. The aim of this experiment, we want to observe osmosis. Now, osmosis is the process by which water moves from an area of high concentration of water to an area of lower concentration of water through a cell membrane. The potato cells have cell walls, yes, because they are plant cells, but underneath the cell wall there is a cell membrane, and the water molecules would move along their concentration gradient, the concentration gradient of water. So it's moving from where water is in higher concentration to where water is in lower concentration. So what we aim to do with having put our potato strips into tubes containing different concentrations of solution, we want to see whether there's going to be any difference in the end point. So when we come back and we do our measurements, we should then be able to see whether or not our potato strips have grown larger, or whether they have shrunk a little bit, or whether they have remained the same size. And that will enable us to make some sort of concluding statement about osmosis taking place with our potato in these different solutions. So we've come back now to look at the results of our osmosis investigation using the pieces of potato. We had weighed and measured 2 dimensions of each of our potato strips, and those can be seen written on the covers of the Petri dishes next to each set. Remember that we had 3 sets. In this row, we have potato strips that had been placed into the concentrated solution, salt solution. In the middle, all of these strips were placed into the dilute salt solution, and these strips at the bottom were all placed into distilled water. The original dimensions at the start of the investigation are written for each one of the tubes on their Petri dish covers next to where you're seeing the potato strips. So what we are observing now is the result after the strips had been left soaking in the solutions for approximately 1 hour. You should be able to observe that the strips that had been placed into the distilled water are now quite a lot larger than they were originally. These are the original dimensions, and we are going to show shortly a table in which the measurements have been compiled. But for now, you want to look at the differences between the potato strips that had been placed in the various solutions. The ones which had been placed in the dilute solution appear to be the largest, and at the top row we can see that the strips which were placed in the concentrated salt solution appear to have become a little bit smaller in size than they were originally. The strips in the middle seem to have stayed approximately the same size, but we can tell the difference when we do our calculations using the data that has been written on this sheet of paper. So the results have been collated and are on this sheet, and what you need to do is to calculate the difference in size between the original size of each of the potato strips that had been placed into the 3 different areas, and then calculate the mean change in the size of the strips. Once those calculations have been done, your next job is to write your discussion in which you're going to try to explain the results that you have obtained in this particular investigation. When we meet in our virtual classroom to discuss the lab, we will talk more about the role of osmosis in the life of the plant, and what we were looking at here, how does this investigation really demonstrate that process known as osmosis, the movement of water from areas where water is in high concentration to where water is in lower concentration across cell membranes. And the cell membranes that we use today are those that are found in the potato cells in each of our samples.